Translation of experimental to clinical neuroscience. This is my summary of the Sci 354 course from Macquarie University, Clinical Neuroscience. And to begin with, here are the reasons as to why we use animal research. It allows for systematic manipulation and for scientists to analyze cause and effect. It is easier to conceptualize simpler systems, since using simpler systems are much easier to understand. Rodents have similar neuronal systems, and not all procedures can be performed on humans because it might not be ethical or practical, and thus it needs to be approved by the Animal Ethics Committee to minimize pain wherever possible. Models include the use of rats, mice, zebrafish, and certain monkeys like macaque or rhesus monkeys. So translations are defined as the scientific advances using animals to improve human health. 37% of animal research is translated to humans. This is probably due to the fact that humans have bigger and more complicated systems, and that there's quite a lot of variation between the species. One must also look at the validity of behavior models, since behavior models should be used to accurately reflect the disorder that is being studied. So, for example, one must think about predictive validity, whereby does the model reflect the disorder, and can drugs be used to treat it? Are the drugs comparable? As in, like, if the drugs are used on schizophrenic mice, for example, can it be used on schizophrenic humans? There's also face validity, which is whether the model shows the same symptoms in animals as within humans. So let's say, using that analogy of schizophrenia, do the mice who are made to exhibit symptoms of schizophrenia, are these similar to the ones that are in the human version of schizophrenia? Constructive validity is how well does the test measure what it is supposed to. So this is the construct quality. Moving along, there has been an effect of publication bias, whereby journals only publish and as a result give more funding to positive or significant results. Kellner's presentation in 2012 found that there has been a failure to publish negative results. I'll be talking about similarities and differences in using animal models. A similarity includes that chimps share 96% of the human genome, and that rats share 95% of the human genome. Gibbs et al. In 2004 stated that rats, mice, and humans encode similar numbers of genes, and that almost all genes are involved in human diseases are also present in rats. There are also anatomical similarities of the brain. Differences, however, include that rats and chimps just are not humans. They're not human at all, and that using behavioral or neurochemical models are merely pieces used to understand the disorder. You are unlikely to ever create a complete model of human diseases from animals. Schizophrenia models. So we've been using animal models to model the disease of schizophrenia, the disorder, since the medication that we have been using hasn't really changed in the past maybe 70 years. Medication treats positive symptoms, such as the use of promazine, and that was developed in the 50s. Not much has changed, and also research needs to focus on treating negative, such as withdrawal and cognitive symptoms. There is a dopamine hypothesis whereby schizophrenia is believed to be caused by more dopamine in the limbic system, and thus having less dopamine in the prefrontal cortex, thus causing hyperfrontality, which drives negative and cognitive symptoms. The nucleus accumbens increases dopamine, which causes positive symptoms like psychosis and euphoria in the beginning. So basically, according to the dopamine hypothesis, there's too much dopamine in one area, in the limbic system, and not enough dopamine in the other area, in the prefrontal cortex. Phencyclidine, or PCP, is a NMDA receptor antagonist, which is used to chemically induce psychosis in the mice models. They produce auditory hallucinations, cognitive impairment, and paranoid delusions. It is used to help screen for antipsychotics. So basically, a rat is given PCP, and antipsychotics are added to it to see whether they treat the psychotic symptoms. Matrix, or NIMH, is the measurement and treatment research to improve cognition in schizophrenia. It was developed by the National Institute of Mental Health, or NIMH, and they basically listed 7 deficits of which schizophrenia affected cognitive function. These include working memory, attention, problem solving, social cognition, reasoning, visual learning, and verbal learning. The rodent models are made to be schizophrenic by adding methamphetamine, since that can induce like psychotic-like symptoms, psychosis. And also, it could include, as in to create schizophrenic mice. One could also do it pharmacologically, whereby they change the glutamate NMDA receptors by adding an antagonist to it, or by adding PCP, which we mentioned before, or ketamine or diazepam. One could also change the genetic makeup of the rodent by altering its dopamine and glutamate transmission, or finally they could affect its neurodevelopment, such as through pre or perinatal insult, which is MAM, or the lesion of the hippocampus, or LPS infection in gestation, or via adding a certain polyic acid, or through maternal deprivation or social isolation. After any of these, whether pharmacological, genetic, or neurodevelopmental ways of inducing schizophrenia, we then could place the rodent into a novel object recognition task, whereby rats are exposed to two of the same objects, then exposed to one known object and one novel object. Rats with visual memory will spend more time at the novel object after a 1 to 24 hour delay. There's also a novel conspecific discrimination task, whereby exposure occurs to other rats as opposed to objects, and this helps to check for the rat's social cognition. Rats are exposed to one rat, which is enclosed, then to a novel rat. Rats with intact social cognition should spend more time with the new novel rat, while schizophrenic modeled rats spend less time with others. 5CSRTT, cognitive speed and accuracy. 5 holes fitted with stimulus lights and nose sense detectors are placed into what is similar to a Skinner box. If the rat places its nose into the right hole, as in the 1 of 5, then the room lights up and they get a food pellet. If they suffer from some cognitive or attention deficit, then it is expected the rats may make a premature or omission or incorrect result, which ultimately results in no delivery of food. Nikiforok's PCP model, for example, showed that there was consistent deficits in all 7 tasks of rats which were induced with schizophrenic rodents, but this was only for acute episodes. Heterogenous disorders. These are disorders that manifest in many different ways, which include schizophrenia in the DSM-5, since there's like schizophreniform and schizoid and so on and so forth. So now moving along, I'll be talking about models of post-traumatic stress disorder, PTSD. PTSD is when individuals are exposed to extremely traumatic situations involving war, combat, rape, or sudden loss of family or friends. There are memories associated with fear, and they are hard to suppress and can be triggered via external cues by surroundings at any time, which thus produce severe anxiety for the sufferer. There has been a theory which posts that the HPA axis is to blame, whereby the amygdala and hippocampus drives and affects the hypothalamus, causing the release of corticotropin releasing factor, CRF, into the pituitary gland. As a result, this releases ACTH in the blood and it reaches the adrenal gland, producing cortisol. In PTSD, the amygdala becomes dominating and the hippocampus can't inhibit the ACTH. In their study, the Sloriers et al. 2018 created a post-traumatic stress disorder model based on 4 requirements, whereby it lasted longer than a week after the trauma. The rodent must have greater than 1 behavioral outcome measured related to post-traumatic stress disorder. It must be replicated in other laboratories, and it must use variable stresses to prevent habituation. We must also consider biological phenotypes, which can include neuroinflammation or cell distress, sleep disturbances, as well as the reversal of serotonin reuptake inhibitors, SSRIs. Social defeat stress, SDS. Desloriers et al. 2018 had an experimental mouse with an aggressor mouse for 3 to 10 minutes, and this sensory contact occurred every day. The mouse was separated, and this repeated for a period of 5 to 21 days consecutively. Results indicated that the experimental mouse had avoidance cues, had reduced control of the amygdala by the prefrontal cortex, had increased prefrontal cortical activity with the feet, and had reduced hippocampal volume, as well as increased corticosterone or cortisol in mice. Further studies used unpredictable variable stresses, whereby they alternated as to certain types of stresses given to the rodents. For example, there is daily exposure to stresses like social stress, which we've mentioned before, forced swimming, light or dark cycle shifts, so this changes the day to night time or night to day, thus confusing the circadian rhythms of the animal. There's also damp or no bedding, cage tilting, overcrowding, tail suspension, restraining stress, food or water deprivation, predator scent, thermal stimuli, and inescapable shocks, etc. Results: it was found that the rodents didn't show avoidance of cues when given unpredicted variable stress. However, regardless, these two correlated with trauma, depression symptoms, and increased activity in the amygdala. There were damaged neurons in the prefrontal cortex, an increase in the CRF and corticosterone, and there was a reduced hippocampus. So going back to the aforementioned predator stress, which was from in the UVS that I've mentioned, the rodent is exposed to a cat for 10 minutes and it is unprotected for 10 minutes, or the rodent senses the smell of the predator over a period of 10 days. And results showed that cue avoidance was connected with trauma, but that there was no depression symptoms reduced in the prefrontal cortical / amygdala connectivity. There was also increased corticosterone length and a reduced dendritic length and reduced spine density in the hippocampus. Finally, I'll be talking about models of drug addiction, which includes features of drug reward or altercation. I've talked about this many times before in previous videos, but I'll go over it again. So there's maintenance, which is the drug taking behavior. There is motivation for the drug taking behavior, which can be measured by how many levers that the rodent presses, and via a progressive ratio scale to see how many times they press to get the infusion via intravenous drug administration. There's also extinction of the drug taking behavior, whereby the rodent stops responding to cues, and this is what we are interested in because we desire to treat or cure people of a drug addiction. Relapse is the coming about, reinstating of this drug taking behavior triggered by drugs, cues, or stress. Neurotransmitters translation. Alfama et al. 2018 saw changes in oxytocin neurons. Both humans and rats only had one oxytocin receptor, and oxytocin in rats had a large effect on auditory and olfaction processing. Humans, however, relied on oxytocin usage in their visual attention and gaze for the recognition of conspecifics or threats. We see a deviation in the ability to model as in between rats and humans, since oxytocin systems are most appropriate in stress models for addiction, food intake, and pair bonding. As in, despite these differences, in both rats and humans, we still rely on oxytocin for pair bonding behavior, food intake, and addiction, or used to maintain an addiction in addicts. Ultrasonic vocalization, USV, with behavior is used to hear what rats say to each other and measure their social dysfunction. So for example, 50 kilohertz implies that there's something good happening with the rat, and they produce a trill sound when re-exposed to methamphetamine. They have 22 kilohertz when they are not happy, as in the sound that they produce is at the 22 kilohertz frequency and it is not happy. The reason why we use ultrasonic vocalization is because we can't hear the rats make the sound. Using this equipment, we can then hear what they make, and then we could, as a result, infer their possible emotion in reaction to drugs, for example, or social stresses, or so on and so forth. So in conclusion, we looked at translation of experimental to clinical neuroscience through animal models. We also looked at the application of research of animal models into schizophrenia, post-traumatic stress disorder, and drug addiction. And yeah, join me for my last video whereby I'll talk about different types of neurostimulation. Bye bye.